With the advent of personalized genomics, the incidence of variants of unknown significance, or VUS, in disease predisposing genes is increasing. When no standardized diagnostic procedures are in place to assess pathogenicity of such variants, targeted healthcare cannot be implemented for carriers and their relatives. Lynch syndrome is a common inherited predisposition to colorectal and other visceral cancers that is caused by a defect in one of the genes of the DNA mismatch repair pathway. Mismatch repair is a canonical DNA repair pathway that acts to remove inadvertent misincorporations introduced in DNA during its replication. Here, the replicative DNA polymerase has misincorporated a thymidine instead of a cytosine, opposite a guanine. In wild-type cells, the guanine-thymidine mismatch is bound by the heterodimeric MSH2-MSH6 mismatch repair protein, after which the heterodimeric MLH1 PMS2 mismatch repair protein is recruited. This protein complex initiates removal of the misincorporation containing DNA strand. This is followed by resynthesis by the DNA polymerase, this time without making a misincorporation. In Lynch syndrome, a pathogenic defect in one of the 4 DNA mismatch repair genes results in loss of mismatch repair. This leads to the persistence of the misincorporation and therefore in the accumulation of spontaneous mutations, including a hallmark phenotype called microsatellite instability or MSI. These phenotypes cause a dramatically increased rate of cancer development in Lynch syndrome. Diagnosis of Lynch syndrome is based on clinical criteria and the subsequent search for a pathogenic defect in one of the mismatch repair genes, called step 1. Individuals with a pathogenic defect greatly benefit from lifelong surveillance and may, additionally, benefit from both targeted lifestyle measurements and treatment, whereas unaffected relatives are dismissed from preventive surveillance. In case a variant of uncertain significance is identified in one of the DNA mismatch repair genes, of which pathogenicity cannot be determined, all family members, whether they carry the variant or not, remain suspected of Lynch syndrome, which precludes targeted healthcare and puts a great burden on the entire family and on preventive healthcare systems. Here we propose the development of a standardized diagnostic pipeline for variants in mismatch repair genes as identified in individuals suspected of Lynch syndrome. This pipeline is based on the causal relation between defects in DNA mismatch repair and Lynch syndrome, on a profound knowledge of the genetics, cell biology and biochemistry of DNA mismatch repair, and on the availability of assays to test the activity of the DNA mismatch repair pathway as a whole and of individual components of the pathway. To assess whether a variant of a certain significance is pathogenic, the diagnostic pipeline proceeds to step 2. In this step, the fuse is first analyzed by computer algorithms that are based on comparative analysis of the DNA mismatch repair genes from a wide range of species. Step 2 also comprises a novel, cell-free assay that measures the integral activity of the DNA mismatch repair pathway in a test tube. The complete in vitro mismatch repair assay only requires information on the nature of the variant of uncertain significance from a clinical geneticist. It is performed entirely in a test tube and is exemplified here for testing variants in the heterodimeric MSH2-MSH6 DNA mismatch repair protein. The variant gene and its encoded protein are synthesized in vitro. Next, the variant protein is incubated with an extract from cells that are deficient for MSH2 and MSH6, and also with a DNA substrate that contains a TG mismatch, disrupting a restriction enzyme recognition site and the fluorescent label as represented by the little sun. Repair of the mismatch results in the recreation of the disrupted restriction site. Cleavage of the site results in a diagnostic DNA fragment that can be quantified by automated fragment analysis. Deficiency for DNA mismatch repair results in the absence of the diagnostic fragment and therefore provides strong evidence for pathogenicity of the variant. In some instances, the step 1 and 2 assays may not provide conclusive results as to the pathogenicity of the variant. In these cases, the diagnostic pipeline will proceed to step 3. In step 3, an additional layer of diagnostic assays can include an examination of other cell biological, biochemical and biophysical analyses of the DNA mismatch repair genes and their proteins. These assays often address more specific aspects of the protein function beyond measuring repair of mismatches. The Step 3 assays require specialized expertise, laboratory facilities, and also trained scientific personnel.
One example of a Step 3 assay is shown here. This assay measures correct cellular localization of the variant protein. As shown here, the variant MLH1 protein does not enter the nucleus unlike the normal protein. Therefore, this result supports pathogenicity of the variant. Step 3 assays cannot stand alone, but they can provide additional evidence in favor or against pathogenicity in case the results from the Step 1 and 2 assays are inconclusive.
So how can we use the results from the Step 1, 2, and 3 analyses to define pathogenicity of an unclassified variant in one of the mismatch repair genes? We propose to generate a software-based hierarchical decision tree that, based on the results from the different analyses, guides the clinical geneticist through the different steps of the diagnostic pipeline. The software will evaluate and integrate the results and perform a Bayesian analysis that outputs a pathogenicity probability score ranging from not pathogenic to definitely pathogenic. This will allow the clinician to make rationalized decisions as to the management of the affected individual and his or her relatives. DNA mismatch repair is one of the best studied DNA repair pathways, and biochemical defects in the pathway are causal of Lynch syndrome. Assays are available to study the activity of the entire DNA mismatch repair pathway and of its components. These assets provide a unique opportunity to develop and standardize a diagnostic pipeline for the analysis of the pathogenicity of variants in one of these genes. Such a diagnostic procedure will not only greatly benefit individuals suspected of Lynch syndrome and also their relatives, but moreover provide an example for the translation of genomic data into targeted healthcare in the upcoming age of personalized genomics. More information can be found in our accompanying review in Human Mutation.